So welcome to this video on an introduction to functional neuroanatomy. Today we're going to talk about the general principles of information flow, how information travels through the nervous system so that different parts of the nervous system govern function in different parts of the body. And this pretty diagram you're looking at here is the fruit fly connectome, published in 2023. It's a complete wiring diagram of the baby fruit fly brain, and it's also very beautiful. So it reminds me of the balloons in Up. Kind of makes me happy. Now today we're not going for a complete wiring diagram of the human nervous system. Rather, we want to talk about general principles of connectivity and how we can use that information to link neural control to function and dysfunction. So the paths of afferent and efferent information flow in the nervous system follow a few general principles. Of course, afferent means toward, and in this case the flow is toward the central nervous system, while efferent means away from or out of the central nervous system. And the really important one of the most important principles is that of contralateral control. And that means that most motor and sensory pathways cross the midline at some point to govern contralateral parts of the body. It's just a central premise of neural control. And by the midline, I of course mean this midline, the mid-sagittal plane. So this is a little schematic of someone using their right hand to test the temperature of water in the shower in the morning. And the information from that right hand that originates at sensory endings in the skin ultimately impinges on the left side of the brain. So it shows you here, following the blue sort of path of sensory information takes, crossing because it's temperature information at the level of the spinal cord and ascending ultimately to the thalamus and then the left somatosensory cortex. Similarly, the efferent command originates in the left side of the brain, descends, crosses at the level of the pyramids, and governs musculature in the right hand. So there's a few little weird simplifications in this diagram. Sensory axon enters the spinal cord and synapses with brain. A little bit general. Of course, we know it synapses in the thalamus, but the diagram makes the point nicely. Contralateral control, motor and sensory pathways cross at some point. Before we go further, let's make sure we know what contralateral control and contralateral means and talk about a little bit of other crucial terminology. So ipsa and contralateral. Ipsilateral means same side of the body and contralateral means opposite side. Nucleus, which is singular, or nuclei, which are plural. Nuclei are collections of gray matter in the central nervous system, so in the CNS. We look at the next slide for a second. It's funny when, learning science in English, if you ask a physicist, a botanist, and a neurosurgeon, you'll get three different working definitions of what a nucleus is, right? So the nucleus of an atom, the nucleus of a cell, or a nucleus in the brain. And this is the one that we're talking about. I tried to come up with a joke, a physicist, a botanist, and a neurosurgeon walk into a bar. I didn't get anywhere. So there we are. So this is the nucleus that we're talking about. And any collection of cell bodies in the CNS is a nucleus. You can think of the cranial nerve nuclei. The cranial nerve nuclei reside largely in the brainstem. And of course, they give rise to cranial nerves. And the cranial nerve is part of the PNS. The nucleus or nuclei is part of the CNS. And ganglion, by analogy, or ganglia, those are collections of gray matter in the PNS. And so I said here, what's the exception to this rule? We often use the term basal ganglia. That is actually incorrect in terms of anatomy. We should call them basal nuclei, right? Because of course they're in the CNS. So you'll still hear the term basal ganglia even in a textbook, but basal nuclei is the correct term. So that's the gray matter. Moving to white matter, a tract is a collection of white matter in the CNS. and there's other names for this. The most common name is a fasciculus. And I can't spell fasciculus right now. It's close, may not be perfect. So that's another common name. There's other specific names. Lemniscus is a white matter tract. Really specific names like the internal capsule. That's a specific white matter tract. In contrast, a nerve is a collection of white matter, you guessed it, by the pattern in the PNS. Right? So a bundle of white matter in the PNS is a nerve. One little thing when we talk about tracts, this little subtlety, is a commissure versus a decussation. And so it's a subtle thing. A commissure is a white matter tract that crosses the midline almost horizontally or transversely. So it crosses from side to side, like the one I think of as the corpus callosum. Best example of a commissure. A decussation crosses obliquely. Right? And the one I think of is the decussation of the pyramids. Right? So it crosses the nervous system on an angle. Now, when we talk about gray and white matter, the distribution varies along the rostral caudal axis of the nervous system. Just a good thing to keep in mind. In the cerebral hemispheres of the brain, white matter is deep and gray matter of the cortex is superficial. In the spinal cord, that arrangement is inverted. So the butterfly of the gray matter is deep and white matter is superficial. The brainstem is a bit of a mixed bag. It's interesting. And so you get white and gray matter all mixed up, intermingled. There's not as clear a pattern as there is in the hemispheres above and the spinal cord below. And that makes deficits and damage really interesting and potentially catastrophic in the brainstem. So just something to remember about the organization of white and gray matter. So we said we want to talk about the general principles and organization of connectivity. We really want to apply this information. That's the fun part, right? Why do we care what's connected to what and how? Well, we want to know how to localize damage based on the patient in front of us. So take a set of signs and symptoms and say, I think this lesion is in this part of the brain on this side. And conversely, if we see a lesion, we want to be able to predict the functional outcome. If this part of the brain is damaged, I think this is what the patient's going to look like. And to do this, we need to know two crucial things. The first is the location of the relevant gray matter and receptors and effectors. And the second is the path of the white matter that connects the central gray matter to the receptors and effectors. And if we know those things, we can do a lot of interesting stuff diagnostically. So let's start with the somatosensory system. We've already said that sensory pathways cross the midline, creating a contralateral relationship between body and somatosensory cortex. However, what's really important is that the two main somatosensory systems cross the midline in different places, and this has crucial implications for functional outcome. So here we have really simplified schematics to start us off. The dorsal column system on the left, mediating fine touch, pressure, and proprioception, and the anterolateral system, often referred to as the spinal thalamic tract, or STT, which mediates pain, temperature, and I would argue deep touch rather than light would be my interpretation of that. Both of these systems follow a similar schematic. So they start at a first order neuron. There's a receptor in the periphery, a first order sensory neuron in the dorsal root ganglia, in this peripheral nervous system, a second order neuron that ascends to the thalamus, and from the thalamus, a third order neuron that ascends to the somatosensory cortex. So that's the same for both. What is crucially different is where the sensory information crosses the midline. So in the dorsal column system, it's here in the level of the brainstem, and in the anterolateral system, it's here at the level of the spinal cord. And those two different crossings have crucial implications for function. Both, I've said, impinge on the thalamus. And I just want to take a step back. We often talk about the thalamus as a nucleus. It's actually a big nuclear complex. So it receives all kinds of information. You can see the limbic system, the reticular formation, the cerebellum, basal ganglia, all of these systems impinge on the thalamus, as well as the sensory inputs that we normally associate with the thalamus. And every type of information has essentially its own little nucleus. So if you divide the thalamus up, it's subdivided and subdivided into different nuclei. The thalamocortical pathways that has extensive connections with the cortex tend to be reciprocal. So thalamus to cortex and cortex back to thalamus, chattering back and forth between one another. And they also tend to be uncrossed. So ipsilateral thalamus to cortex pathways. So that's a little digression about the thalamus. The dorsal column medial lemniscus pathway. Let's take it a little bit slower and make sure we know what's what in this pathway. We've got a receptor in the periphery, a primary sensory neuron in the dorsal root ganglion, often abbreviated DRG. That axon continues to ascend. The axon of that primary sensory neuron, here it is, ascends in the dorsal columns ipsilaterally all the way to the medulla, where it synapses on one of two nuclei, the nucleus gracilis or the nucleus cuneatus. That secondary sensory neuron, then originating in those posterior dorsal column nuclei, extends an axon which crosses the midline and ascends toward the thalamus. And so the pathway's in two divisions. That's why it's got this name. The dorsal columns describe the pathway from within the spinal cord, right? Of the primary sensory neurons. The medial lemniscus describes the pathway from medulla to thalamus. So dorsal column, medial lemniscus pathway, often abbreviated like this. And the important point is the information crosses in the medulla. The intralateral or spinal thalamic system follows the same general scheme with primary and secondary neurons, but those neurons originate in different places. So we here have our receptor leading into a primary sensory neuron in the DRG, or dorsal root ganglion. That synapse is typically on a second order neuron in the spinal cord. And that second order neuron crosses at or near the level of the spinal cord, commissurally, right, a horizontal crossing, to ascend in the anterolateral pathway or the spinothalamic tract. And so the place where that crosses is at or near the level of the primary sensory neuron. So what are the implications of different sensory pathways crossing the central nervous system midline at different levels? Well, the deficit pattern is determined by whether the lesion occurs above or below the point of crossing. And this is a question you want to ask yourself for every pathway you're studying in functional neuroanatomy. Is the lesion above or below the crossing point? So in this little schematic, we can use that to talk about a thought experiment. We'll assume that it's in radiological orientation, right? So what does that mean? It means the patient's sort of facing me. So the left side of the image is the right side of the patient, and the right side of the image is the left side of the patient. And in this schematic, the spinal thalamic tract is blue. The dorsal column medial lemniscus pathway is green. So if we put a lesion here, what would the deficit pattern look like? And if we put a lesion here, what would that deficit pattern look like? And so you want to be able to describe the deficit pattern in terms of ipsilateral or contralateral and the type of information loss. So that's a little challenge. So let's switch gears for a second and go to the cortical spinal tract, abbreviated CST. And I want to say right off the bat that the cortical spinal tract is not the only descending pathway for motor control. So there's a whole bunch of what we call extrapyramidal tracts that crucially govern movement. For now, we'll focus on the CST, but is not the only tract involved in motor control. The CST also governs the contralateral side of the body, and this is the pathway that the most of the axons in the CST take. So they originate here. You have an upper motor neuron originating in the motor cortex, the precentral gyrus. Those axons descend via the internal capsule, cross at the level of the pyramidal decussation, so that's an oblique crossing, and descend in the lateral corticospinal tract to innervate a lower motor neuron in the ventral horn. And that, of course, extends to innervate skeletal muscle ipsilaterally. Now, I said most of the axons cross in the decussation. There is a small component of the anterior corticospinal tract that doesn't cross here in the pyramids. It descends ipsilaterally, but these axons can't cross at the level of the spinal cord. So they still facilitate contralateral control. So that's a subtle difference. This small component that descends past the pyramids ipsilaterally, but still governs the contralateral side of the body. Now, another thing we tend to forget about when we talk about motor control, everyone thinks of the corticospinal tract. We forget about the cortical bulbar tract. So the cortical bulbar tract is analogous to the corticospinal tract, but governs cranial nerve nuclei and therefore muscles of the head, face, and neck. Now, it follows a similar pattern. So if we think about the corticospinal tract originating here, the cortical bulbar tract originates more laterally in the head and neck area of the primary motor cortex, descends via the internal capsule, and then peels off in the brainstem all the way down to innervate the cranial nerve nuclei. And cranial nerve nuclei are a bit more complicated. Many of them are innervated by both crossed and uncrossed fibers from the cortical bulbar tract. So there isn't as clear a pattern of contralateral control. There's a bit more redundancy. There's still some dominant contralateral control, but not perfect contralateral control. Now let's just talk a little bit about basal ganglia and cerebellar circuits. So these are feedback loops that, these structures are involved in feedback loops that modulate motor control. So they govern motor control indirectly. They do not have direct projections to lower motor neurons, but they do send input to the primary motor cortex via the thalamus. And this input tends to control movement via feedback loops. So this is what you're looking at here. In red, you're seeing a feedback loop between motor cortex and pons. I know I'm drawing red on red, but we're doing the best. And cerebellum and cortex, right? That's the thalamic feedback loop, if you will. In the blue, you're seeing feedback loops between motor cortex, basal ganglia, and thalamus. And so via these feedback loops, the cerebellum and the basal ganglia indirectly modulate movement, but they do it in slightly different ways. Now, this is a complicated slide, and you don't need to know all of the details yet. But what you do need to know is that the cerebellum is sort of a movement adjuster, and as such, it receives a lot of sensory input. Sensation of different parts of the body typically is represented ipsilaterally in the cerebellum, so ipsilateral between body and cerebellum. But because cerebellar outputs are directed toward motor cortex via the thalamus, cerebellar damage causes problems with movement, not sensation. So yes, it receives a lot of sensory input, but the deficits associated with cerebellar damage are movement deficits. They also tend to be ipsilateral deficits. So damage to the cerebellum indirectly affects movement of the ipsilateral side of the body because of the connectivity pattern. Now, conversely, while the basal ganglia also indirectly affect movement, they work via a different mechanism. So basal ganglia are involved in planning movement, but not adjusting. So they don't receive the direct sensory input that the cerebellum does. Because the basal ganglia largely connect to and influence motor cortex in the same hemisphere, damage to the basal ganglia affects movement on the contralateral side of the body.